Alright guys and gals, let's continue our discussion of the cell cycle. We just finished up talking about mitosis, and let's move on now to focus on meiosis. Meiosis can often be described as, and I've often described it to students as, mitosis happening twice in a row, 2 rounds of mitosis. Ultimately, with meiosis, you'll take a you'll take one diploid cell and you'll turn it into 4 haploid cells. In other words, you'll go from 1 nucleus to 4 nucleus. You'll go from 1 cytoplasm to 4 cytoplasms. The reason we have meiosis take place is so that we can cut the number of chromosomes in half, so that we can make sex cells that have half the number of chromosomes and recombine them to the correct number of chromosomes for a fully functional human being. If it wasn't for meiosis, there would be a doubling of the number of chromosomes in human beings with every generation, which would effectively kill off the human race within a single generation. The first half of meiosis is when we go from 1 cell to 2 cells. This is known as meiosis 1. And meiosis 1 has stages that are very analogous to mitosis, hence the name prophase 1, metaphase 1, anaphase 1, telophase 1. During prophase 1 in particular, we're going to have synapses occur. And during synapses, we're going to take homologous chromosomes and line them up right next to each other. So 2 chromosome number 1s, 2 chromosomes number 2, they'll be lined up side by side with each other. And during this stage, during synapsis of prophase 1, when these homologous chromosomes line up with each other, we can have crossing over occur. Crossing over is shown right here in this figure, where half of a piece of one chromosome, one sister chromatid, part of a sister chromatid is being swapped with another sister chromatid. Then we have metaphase 1. Metaphase 1 is when the homologous pairs line up in the equator. Anaphase 1 is when the homologous chromosomes separate from each other. And then telophase 1 is when we have 2 daughter cells that result. So here we have prophase 1. We have the homologous chromosomes pairing with each other. And then during metaphase 1, the homologous chromosomes have had crossing over already occur, and now they're lining up in the middle of the cell. During anaphase 1, they're being pulled apart. And notice how these duplicated chromosomes still have their centromeres in the center. And then at the very end, at telophase 1, after pulling those chromosomes apart, we will have 2 separate cells, each with duplicated chromosomes in them. In between meiosis 1 and meiosis 2, there's a stage known as interkinesis. Most textbooks don't focus on this very much because interkinesis is not a very long stage. Now we need to talk about crossing over. Crossing over occurs during prophase 1 of meiosis 1, and this allows for mixing of genetic information between separate chromosomes. This mixing of the genetic information increases genetic variation and is a source that minimizes mutation rates among human beings. So here we have 4 sister chromatids on 2 duplicated homologous chromosomes, and we can take different alleles of the same gene and swap them with each other to make a new chromosome by mixing and matching pieces of adjacent chromosomes with each other. So part of chromosome 2 swapped with chromosome number 3. Something else that's worth emphasizing is we also have some variation occur from independent alignment. When we're lining up chromosomes during metaphase 1, there's no hard and fast rule that says that all the maternal chromosomes need to be on one side and all the paternal chromosomes need to be on the other side. Instead, for chromosome number 1, you flip a coin. There's a 50-50 chance that mom's chromosome will be on one side and the paternal chromosome will be on the other side. And this repeats for each chromosomal pair. And this only is going to dramatically increase the possible number of genetic combinations the gametes form. You can have all the maternal and all the paternal chromosomes on one side. You could have 3 and 1. You can have, 2 and 1. You can have a mom, dad, mom. You can have a dad, mom, mom. You can have a dad, mom, dad, 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 mom, mom, dad, dad, so on and so forth. You get the idea. There's many different ways you can mix and match chromosomes. And with just 3 pairs of chromosomes, we have 1, 2, 3, 4, 5, 6, 7, 8 combinations. That's 2 times 2 times 2. Now, if we had 23 pairs of chromosomes, which we do as human beings, we can take 2 to the, and I'm using my calculator for this, FYI, to the 23rd power. Because of independent alignment, this is going to cause us to have 8.3 million new combinations of chromosomes in our gametes. Never mind all the different combinations that we get from crossing over. So after meiosis 1, we now are going to have meiosis 2. During meiosis 2, we'll have prophase 2, metaphase 2, anaphase 2, and telophase 2. And during these different stages, we're going to pull duplicated chromosomes apart. So during meiosis 1, meiosis 2, we are going to enter prophase 2 where the chromosomes condense, and then those chromosomes are lined up in the midline. This is going to look very similar to mitosis. In meiosis 1, let's back up here. In meiosis 1, you had two pairs of chromosomes, or two homologous chromosomes lined up in the middle during metaphase 1. During metaphase 2, you have single chromosomes being lined up in the middle of the cell. And then those single chromosomes are pulled apart, the identical sister chromatids are separated, and you have daughter cells form, which each have one set of DNA. And these daughter cells are haploid in nature because they have one set of DNA. This process is used to make sex cells, so both the sperm and the eggs are made. The process of making sperm is known as spermatogenesis, and this is how we make sperm in males. This is a continuous process that takes place during the entire lifespan of the male and takes place within the seminal tubules inside of the male's testes. This process results in about 4 million sperm being produced every single day. Most of those sperm are going to die and be reabsorbed into the body. Oogenesis is the process of making an egg. We still have the same root word here, genesis, to create. And in females, this process is a little bit more convoluted. In males, during spermatogenesis, they start with one parent cell and end up with 4 identical sperm cells. So we have one parent cell that ends with 4 identical sperm cells. In oogenesis, we have one really large primary oocyte that turns into one really large secondary oocyte that then is paused in the middle of meiosis 2. And meiosis 2 does not finish until a sperm enters the secondary oocyte and finishes meiosis 2. For a very brief period of time, we technically have an egg, and once the sperm's nucleus and the egg's nucleus have fused, fertilization has occurred, and now we have a zygote instead of an egg. Those polar bodies are genetic leftovers from the process of making the secondary oocyte. These polar bodies are small capsules that contain excess and leftover genetic material. So during spermatogenesis, the genetic information is divided 4 ways in 4 equal cells. During oogenesis, the genetic information is still divided 4 ways, but it's not going to be divided 4 ways in 4 equal cells. Most of the genetic information is going to be discarded in these polar bodies. Only some of the genetic information, 1/4 of the genetic information, makes it into the resulting egg. We also need to emphasize that only 1 egg is produced per month during the female's reproductive lifespan, and typically there will be 500 eggs produced, or 500 secondary oocytes produced during the entire reproductive lifespan of a female. And if we think of it as 500 oocytes, we think of it as approximately 0.9 months per oocyte times 500 divided by 12, we're looking at about a maximum of 37.5 years, or a maximum of 40-ish years where a woman's body is fertile. And I think that's a very liberal estimate, according to my back-of-the-napkin math. Let's move on. Guys, gals, how does meiosis contribute to genetic diversity of genetic diversity among the gametes? Go ahead and pause this video right now to get me an answer. 5, 4, 3, 2, 1. All right, the best answer is crossing over, as we mix and match between maternal and paternal chromosomes, but independent alignment also does that as well. So the correct answer is more than one of the above. We have both crossing over and independent alignment that increase the genetic diversity of our gametes. Boom. Let's move on and compare and contrast mitosis to meiosis. Mitosis, generally speaking, is going to be to generate new cells to repair damaged tissues, whereas meiosis is to make our sex cells, our sperm or our eggs. Mitosis starts with one parent cell and results in two daughter cells, so it only needs one round of division, whereas meiosis starts with one parent cell and ends with four daughter cells, so we need two rounds of division to give us the 4 cells at the end versus the 2 cells at the end. It's also worth emphasizing that during mitosis, the daughter cells from mitosis are diploid, whereas the daughter cells from meiosis are haploid. Here's another figure to compare and contrast. During meiosis 1, we are going to have homologous chromosomes lined up adjacent to each other, whereas in mitosis, those we are going to have duplicated chromosomes lined up single file in the middle of the cell. And then if we compare meiosis, or the end of meiosis to mitosis, we have the 4 haploid daughter cells from meiosis and the 2 diploid daughter cells from mitosis. It's also worth emphasizing that occasionally mistakes happen. Nondisjunction is a mistake that occurs when the homologous chromosomes do not separate from each other appropriately or evenly during meiosis. So in other words, the homologous pair of chromosomes goes to a daughter cell. This results in a gamete that is either +1 chromosome or -1 chromosome. If the gamete is minus a chromosome, that child could then have a monosomy, where instead of having a pair of chromosomes, one from mom, one from dad, instead that child could only inherit one chromosome from the mom-dad couple. A good example of that is Turner syndrome. If a child only inherits an X chromosome from mom or dad, one X chromosome, so it's to be an XX or XY, they're just X single X. That is known as Turner syndrome. And since we only need one copy of the X chromosome to be living, Turner syndrome, generally speaking, is a relatively mild syndrome. And that's only because of X chromosomal inactivation. Other chromosomal defects where there's an extra chromosome are called trisomies. Most trisomies are fatal and result in miscarriages. Occasionally, for example, with Down syndrome, trisomy 21. The person can live, but they'll have significant physical, and cognitive setbacks or defects. Here's a figure showing us how non-disjunction can occur. For example, it can occur in the oocyte, where instead of the chromosomes separating, the chromosomes stayed paired together, and that results in an extra chromosome in the egg, and then the child could have a trisomy, or the child could have a monosomy. And depending on when non-disjunction occurs, that also influences the number of gametes or the number of chromosomes that make it into the resulting cells. Non-disjunction can happen in meiosis 1, and it can also happen in meiosis 2. The earlier it happens, the more gametes will be affected. If we look at a woman's body, generally speaking, will have 2 copies of the X chromosomes, whereas the male will have an X and a Y. And because we have duplicate chromosomes here for the sex chromosomes, females are going to need a way to deactivate their extra X chromosome. Males do not have to worry about this because they only have 1 copy of the X chromosome, but for females, one of the X chromosomes are going to be deactivated very early on in development. This deactivated X chromosome forms what is known as a bar body in each of the female cells. Here's an example of someone who has Down syndrome. Down syndrome usually is going to be characterized by mild to moderate physical defects, a slightly shortened lifespan, and will also result in someone having an extra copy of chromosome number 1. And within chromosome 21, that one that they have the extra copy of, there is a gene that's been specifically associated with Down syndrome, causing the symptoms of Down syndrome, and then the GART, the GART gene, that's been associated with causing some of the symptoms of Down syndrome. Some other syndromes are Turner's. Turner's is when we have only 1 X chromosome. We can also have Klinefelter's. This is when this is someone who's a male that has an extra X chromosome. These individuals are going to have underdeveloped sex organs because they're going to have some male response but also some strong female response. Individuals that have Turner's syndrome are typically going to have underdeveloped sex organs. They'll be short, broad shoulder, and have thick folds of skin on their neck. We can also have females that have extra copies of the X chromosome. So there could be 3X, trisomy for the X chromosome, or quadrisomy. Individuals that have trisomy, generally speaking, are going to be okay. They'll have mild cognitive defects, and they'll be tall and thin, so they'll be fairly attractive by most standards. Generally speaking, these people can live regular productive lifespans. Individuals that have 4 copies of the X chromosome, generally speaking, are going to suffer from very severe mental retardation and are going to have significantly shortened lifespans. There's also Jacob's syndrome, having an extra copy of the Y chromosome. These individuals, generally speaking, will be very tall, have a lot of acne associated with hormonal imbalances in their body, and then finally have cognitive problems related to both speech and reading. We can also modify the structure of a chromosome to cause changes within the person's genome. We can lose a chunk of the chromosome. We can take a chunk of the chromosome, and that's known as deletion. We can take a chunk of the chromosome and move it around within the body from one chromosome to another chromosome, or within a chromosome, that's known as translocation. We can duplicate a piece of the chromosome multiple times, or we can flip-flop a chunk of the chromosome, and that's known as inversion. So here's an example of deletion. There was a gene A, B, C, and this chromosome had gene A removed from it. So now the chromosome is shorter and just has genes B and C on the top. You can duplicate a section. So we go from A, B, C, D, E, F to A, B, C, D, E, D, E. So this chromosome's been extended. There's also been an inversion. C and D have flip-flopped with each other, or we can have a translocation. We can have we can start with a P, Q, R on the right chromosome and an F, G, H on the left chromosome, and we can flip-flop those sections. So the Q and the R can go to the left, and then the G and the H can go to the right. During inversion, we're going to need to snip a chromosome and then reattach the broken fragments. So we'll take there will be a gene, generally speaking this is going to be referred to as a restriction endonuclease, that can snip a chromosome and then exchange portions of a chromosome between 2 adjacent chromosomes. And this can result in deletions and possibly duplications depending on which genes the chromosomes are receiving. When we have our deletions occur, we lose genetic information. When we have translocation occur, we are going each chromosome will gain and lose genes respectively. So, guys, gals, I have a concept check question for you. Generally speaking, what happens if there are too few or too many chromosomes in the fertilized egg, also known as the zygote? Will there be a child born with chromosomal abnormalities? Will there be a miscarriage? Almost nothing, because, you generally speaking don't notice it, or none of the above. Go ahead and pause the video and get me an answer. 5, 4, 3, 2, 1. The correct answer is there's a miscarriage. Now, I know that we talked about a lot of the things, a lot of the potential diseases you could see here in individuals and human beings, which may have biased you towards answering letter A. But the reality of the situation isn't. The situation is most chromosomal abnormalities result in a miscarriage in utero, and those individuals never make it to childhood or adulthood. The exceptions that we talked about are the rare exceptions when there can be a chromosomal abnormality and still result in a viable human being. If you have any questions about this material, please feel free to post it on the discussion board, shoot me an email, or swing by my office when you're on campus. Happy studies!